Hello everyone, I'm Janine Cortiguera Baguhin from the Ecosystems Research and Development Bureau of the Department of Environment and Natural Resources. I have a few questions for you. Do you know how to clone plants or make an exact copy of them? Have you heard of the terms plant tissue culture or micropropagation? And how does this method help in conserving our important plant species? Then this video is probably for you.
According to the World Health Organization, 80% people worldwide are more dependent on culturally familiar, simple, and financially affordable herbal medicinal products as a primary source of health care. 40% of the millennials also prefer natural and organic health and beauty products, thereby contributing to the market growth. Do you know what Gugo is? Gugo is a medicinal plant found in warm tropical Asian countries. The species is usually found at low and medium altitudes of the forest in the Philippines. It is a large, woody branch, evergreen climbing vine. Gugo offers many health benefits. It has anti-diabetic, anti-cancer, and antimicrobial properties. The species is also used as treatment for wounds and dandruff, joint and muscle pains, ulcer and abdominal spasms, and respiratory ailments. Gugo is used extensively in the Philippines and other Oriental countries as shampoo. It is also a commercially important forest vine. It is used as raw materials for basketwares and novelty items. It has great potentials for the export industry. However, Gugo is only propagated by seeds, which may take 1 year or more to naturally germinate. The species was never cultivated. The destruction of its tropical evergreen habitat and unscientific overexploitation of its plant parts resulted to its decreased population. Gugo is under severe threat. To augment the demand for Gugo planting materials, an alternative propagation method needs to be developed. And this is plant tissue culture. Plant tissue culture or micropropagation is widely used and becoming more popular for the large-scale production of endangered, threatened, rare, and economically important plant species. This method is the growing of cells, tissues, or organs from juvenile and mature plants on a defined artificial medium in the absence of microorganisms and under controlled conditions. Plant tissue culture is more preferred due to its ability to multiply species rapidly and uniformly, disease-free plants all year round. It is generally used for crop improvement, long-term germplasm conservation, production of secondary metabolites, gene transformation, and genetic engineering. The Ecosystems Research and Development Bureau, research arm of the Department of Environment and Natural Resources, conducted tissue culture study on Gugo. The ERDB researchers took the opportunity to conserve this valuable medicinal plant species. In the study, the researchers were able to determine the best type of explant that is responsive to plant tissue culture, the effective concentration of sterilants and disinfection duration, and the type, concentration, and combination of plant growth hormones which produce the maximum number of shoot buds. One of the significant findings of the study was that the germination period of Gugo was reduced from 1 year to only 10 to 14 days. Although shoots and roots were not fully induced in this experiment, it demonstrated the potential of Gugo to form new tissues and develop shoot buds in vitro. Thus, the need for further or follow-up study in these aspects. If the protocol will be fully developed, we will have sustainable source of planting materials, more Gugo products, more and better jobs for the Filipinos.
In this regard, ERDB will continue its efforts to develop this technology and will continue conserving valuable forest species through plant tissue culture. This has been Janine Cortiguera Baguhin. Let's multiply Gugo. Let's use natural and organic Gugo. Let's go, Gugo!